Good afternoon everyone, especially to our classmates and to our Sir Roger. So I'm here to represent my presentation. So my title is Spawning, Fecundative, and Fertility of Green Mussel Penabilities about ambient temperature 30 degrees Celsius. First is introduction. So Pernabilities is an economical, important marine bivalve native to the tropical Indo-Pacific regions. So that sample is came from New Guinea, Taytay, Palawan. So these pictures of map is I came conduct my thesis in Binduyan Marine Research Station here in yellow, and that mark is the place of Binduyan. So the objectives is to determine the fecundity of green mussels at different temperature levels about the ambient temperature 30 degrees during the conduct of the experiment. So I use different temperature for spawning. So it determines the fertility of rate of green mussels at different temperature levels about the ambient temperature 30 degrees Celsius during the conduct of the experiment. So dito po malalaman kung masusurvive po sila sa tatlong treatment. Okay, ito sa broodstock collection. So, mature green mussel broodstocks were bought from New Guinea, Taytay, Palawan, and transported to the hatchery using a styrofoam box with seawater with cloth to avoid desiccation that may lead to spawning during the transfer. So, pagdating po pala ng sample namin po is, chinek po namin agad yung temperature niya, and then, nakabalot na po siya ng towel na may basang dagat po. So, upon arrival in the hatchery, mussels will clean and condition overnight in 250 L rectangular spawning tanks with seawater and aeration. So, yun po, nilinis po namin yung samples, and then, in overnight siya sa tank na meron na siyang seawater and aeration. Okay, so this table has used 3 treatments. So the first is 30 degrees Celsius, 32 degrees Celsius, and 34 degrees Celsius. So my replicate is I use 9 replicates, and each one is 3. And then the temperature I use 30, 32, and 34 degrees Celsius. And so the water salinity I use 35 ppt. So the preparation of materials. So first I clean the materials that we need in spawning. So there I prepare, and then I prepare the milk tea with water 200 liters, and then the tanks with complete check of temperature and salinity. So this picture has shown that I used in materials in my conduct here, and then randomized setup. So I use draw lots if kung ilan po masusurvive po sa 9 treatments na yon na ginamit ko po sa vaccine. So, the spawning and fecundity. Spawning in activity started through desiccation of green mussel. Water was removed from 30 minutes to 1 hour. The following heater are set up to heat chlorinated seawater for sudden temperature changes height as 15 degrees Celsius for 30 minutes. So, then 25 individuals of green mussels randomly stacked in different basins with 30 degrees Celsius, 32, and 34 degrees Celsius. So, the ambient temperature during the conduct of the experiment was 30 degrees Celsius, and then this temperature served as a control treatment. With the succeeding treatments, heat chlorinated water is added in 20 L water in basins for the higher the temperature to get reach the right temperature. So this picture, the first is chinik ko po yung temperature niya, and then next naman po is nilagyan ko ng heat water para makuha ko po yung right temperature niya in each treatments. Then, ito, yan, pag natapos ko na yan, na i-check yan, i-prepare ko na po yung aeration with hose din po, yan, sa mga 9 basins, and then ito, mag-observe po, mag-spawning, and then ito po yung kanyang pictures niya, parang, pag-spawning pa lang siya, bubula yan sila. Yan po. It means start na sila mag-spawning. So, fertilization. After an hour, the egg and sperm are collected. Sperm cells will combine with eggs at ratio of 50:1. Within an hour after release, in the mixture was left to fertilize. For the fertilization rate, 50,000 sperms and 1,000 eggs in one treatment were combined in one container within 1 hour to fertilize. After fertilization, count fertilization and unfertilized egg with the aid of the microscope. Okay. So this picture, I show that fertilized eggs after 1 hour of mixing in this microscope. And then, yan, I put these samples na nag-spawn dito sa milk tea. And then ito, minix ko yung unfertilized and fertilized egg. And then I count them on microscope with unfertilized and fertilized egg. Okay. So this one, were in record for a use of sample. Tinimbang ko po sila and then yon sinukat ko yung length, width, and height niya po ng samples ko. And then, this picture, okay, this white, green mussel is a male, and the orange is a female, Yeah. So, this one, this is the pictures, green mussel, sperm white, and then orange is a green mussel egg. Yan. So, these results, dito po naman sa fecundity ko. So, dito naman po, sa, sa fecundity ko po, yan, range po sila ng 1,079 to 1,369, na ito po sila ay with significant po sila. And then, pagdating to sa female, yan, range 82 to 82, so hindi sila not significant po. So the fertilization rates, so ito po yung treatments yan ng 13, 32, and 34. So dito did not spawn sa first treatment, second treatment may data po ako ng 8.08 mean, and the standard deviation is 3.95. And then the next treatment is 2.68. And then the standard deviation of 2.73. So, will not significant po siya. So, recommendation, this study successfully tested the effect of temperature shock in the spawning performance of green mussel variation in terms of different temperature of levels up to 34 degrees Celsius. It can be concluded the hatchery production of green mussel is still possible using temperatures up to 34 degrees Celsius based on fecundity. Although fertilization rate decreased significantly with temperature was increased from 32 to 34 degrees Celsius. It is recommended that a further test be made at higher stock density to ensure presence of both male and female green mussel in the spawning tank. So, I recommend that I use a high temperature like 32 to 34 kasi mas madali po silang mag-spawning than the ambient ng 30 is medyo warm sila and hindi po sila masyado magpapaanak. So, yun lang po, and I thank you.